I'm going to demonstrate trial 2 in lab 12, which is the meiosis lab. You guys did not have, this was not required, but I thought maybe if you could see how to do the poppet beads that it would help some of you. So I have set up the poppet beads. Now I'm using the same set of chromosomes, the same 4 chromosomes that I used to do the mitosis simulation. But before I go into meiosis, I want to explain to you a little bit of heredity. Okay, so you can see that beside each of these chromosomes, I have letters, B's and T's. So a chromosome contains strands of DNA called genes, and each gene codes for a particular trait. So we're going to pretend that the bottom 2 beads of each of our chromosomes, the bottom 2 beads represent a gene that I have shown here by a capital B. So for this large green pair of sister chromatids, this large green chromosome, both of those bottom 2 beads are going to be the gene for brown eyes. Capital B is going to represent brown eyes. Now the bottom 2 genes of the homologous pair of the other one are going to have to be for eye color, but they don't have to be the same color. So these are going to be the gene for blue eyes. That's easy to remember because this is a blue chromosome. So big B brown eyes, little b blue eyes. Now notice the sister chromatids do have to have the very same version of that gene because they're identical. They are just a duplication. They duplicated during this single stranded chromosome duplicated during the S phase of interphase, and it's an identical copy. So whatever version of the gene that that one chromatid has, the other one has to be identical. Okay, so we've got 2 sister chromatids with big B brown eyes, and then we've got 2 sister chromatids with little b blue eyes. And then down here, we have another trait that I'm going to have to show you probably. It's a thumb trait in humans. Now, if you look at my thumb, if I hold it right, okay, my thumb is called straight thumb. And straight thumb is represented by a little t. So over here, the blue chromosome, the bottom 2 beads are going to be for straight thumb. Now, if you have the dominant trait actually is when your thumb is all bent back. When you hold your thumb up and it bends back, it's called hitchhiker's thumb. Okay? So if you have hitchhiker's thumb, that's a capital T. So the bottom 2 beads on the small green chromosome are going to represent hitchhiker's thumb. Now, the possibilities that we could get in the sperm or the eggs are going to be combinations of eye color and the thumb trait. All right. So looking here, if during meiosis, this is how the homologous pairs have matched up. If during meiosis, the 2 daughter cells that form, let me erase the letters now, hopefully you can remember. The daughter cells that form during meiosis are going to, the first 2 daughter cells are going to have 2 chromosomes in them each. So the chromosome number is halved after meiosis 1. And then after meiosis 2, it's going to be, oh, didn't mean to do that. It's going to be like this. It's going to have 4 daughter cells that still have 2 chromosomes each. They're just not duplicated chromosomes. So let me move this one over a little bit. All right. So what we have then is we have, if these were sperm, one of the sperm would have, let's see, brown eyes and straight thumb. And this would be the same, brown eyes and straight thumb. And then the other 2 sperm would have blue eyes and hitchhiker's thumb. Okay, So what if they lined up a different way at the beginning of meiosis in metaphase 1? What if they lined up like this? With both of the blues on the same side, what we're going to end up having are four. I won't break them apart again because you know these two will be the same and these two will be the same. So we're going to have 4 cells, and 2 of them are going to have the brown eye gene and the hitchhiker thumb, and the other 2 are going to have the blue eye gene and the straight thumb. Okay, so this is one way that we can get different combinations of traits in the sperm or the egg, and that's one way that we get genetic variation. That happens during metaphase 1 of meiosis. So now let's come back and let's simulate meiosis. Okay, how did I have it? All right. Okay, so what's going to happen is first in meiosis, we're going to have prophase 1, and that's going to be when the homologous pairs find each other. They pair up. That is called synapsis. And what forms is called a tetrad because there's 1, 2, 3, 4 total sister chromatids, and tetra means 4. And then, so that happens in prophase 1, this process called synapsis. And during synapsis, because these 2 chromatids from the 2 separate chromosomes are so close to each other, sometimes they actually exchange pieces of their chromosome. That's called crossing over. And I'm going to do that in both sets. Now, during metaphase 1, the homologous pairs line up at the metaphase plate. So this is different from metaphase of mitosis. During metaphase 1, they line up in pairs, not in single file. So the homologous pairs line up at the metaphase plate. In anaphase 1, the homologous pairs separate, not the sister chromatids. So we have the homologous pairs separating in anaphase 1. And then they reach the opposite ends of the cell in telophase 1. There is a brief period called interkinesis, and then we start with meiosis 2. In meiosis 2, we have 2 daughter cells with only 2 sets of chromosomes. So the parent had 4, now we have the haploid number, which is 2. So what's going to happen is prophase 2. The chromosomes are visible, basically. The spindle starts appearing. And then in metaphase 2, the chromosomes will line up at the middle, the metaphase plate of the cell. They will line up. They will line up at the metaphase plate. This is metaphase 2. And then in anaphase 2, the sister chromatids will separate. Not the homologous pairs, but the sister chromatids will separate. And so let's see what possible combinations of genes we have in the sperm now because of crossing over. All right. Gosh, this is hard to remember. I know this one is blue eyes. So we have blue eyes, and then the bottom 2 beads are blue in this one. So that was straight thumb. So one of the sperm could have big B little t, which is blue eyes and straight thumb. The other one has the bottom 2 beads are green, so that would be brown eyes. And the bottom 2 beads in the second chromosome are green, so that would be hitchhiker's thumb. So that's a whole different combination. And then we can have blue eyes and hitchhiker's thumb. or we could have brown eyes and straight thumb. These are 4 totally different combinations. 1, 2, 3, 4 totally different possibilities in the sperm or the egg that are produced by meiosis. After meiosis 2, you have 1, 2, 3, 4 haploid cells. And because of crossing over, and because of the independent assortment during metaphase 1, the way the homologous pairs line up during metaphase 1, we have genetic variety. So I hope this kind of helps you with the meiosis.